Hello, I'm Ashwarya Devangan and I'm going to present on the topic osmometer. So, what is an osmometer? An osmometer is a device for measuring the osmotic strength of a solution, colloid, or a compound. There are mainly three types of osmometers: freezing point osmometer, vapor pressure osmometer, and membrane osmometer. Let's first look at freezing point osmometer. It measures the depression in freezing point caused by solutes. Now it's working. The sample solution whose osmolarity is to be measured is prepared. This could be a solution of an unknown concentration of solute such as salt or sugar dissolved in a solvent like water. We have to make sure the solution is homogeneous, meaning the solute is completely dissolved in the solvent. The sample of the solution is placed into a small sealed sample chamber of the osmometer. The volume used is typically very small, a few millimeters or less, to ensure quick and accurate freezing point measurements. The chamber usually contains a small sensor that detects temperature changes as the solution is cooled. The osmometer starts cooling the sample at a controlled rate. This cooling process needs to be uniform to avoid introducing errors in the freezing point measurement. If the solution cools, the solute particles in the solution interfere with the formation of the solid ice structure. This is because the solute particles lowers the freezing point of the solvent, which is also known as the freezing point depression. As the temperature drops, the solution reaches a point where ice begins to form. This is the point where the solvent starts freezing and the remaining solution becomes more concentrated with solute particles. The osmometer uses a sensor, typically a thermistor or a similar device, to detect the exact temperature at which the freezing starts. The sensor continuously monitors the temperature of the sample during the freezing process. The freezing point of the solution is recorded by the osmometer. It is the temperature at which the first signs of freezing are detected. In the presence of solute particles, this temperature will be lower than the freezing point of the pure solvent. For example, water freezes at 0 degrees Celsius, but the solution will freeze at a lower temperature. The difference between the freezing point of the pure solvent and the solution is known as the freezing point depression. The freezing point depression is directly related to the osmolarity, concentration of solute particles of the solution. The greater the depression, the higher the concentration of solute particles. Now its applications. There are various applications. Freezing point osmometer, used to calculate the osmolarity or molality of a solution by measuring the freezing point depression. This is crucial in chemistry, biology, and medicine. It ensures the correct concentration of active ingredients in drug formulations. Accurate osmolarity is important for injectable drugs, as incorrect osmolarity can lead to adverse reactions. It is used to measure the osmolality of biological fluids such as blood, plasma, urine, and serum. This helps in diagnosing conditions like dehydration, kidney function disorders, and electrolyte imbalances. Used to measure the concentrations of solutes in water samples, such as in monitoring pollution levels of salinity in oceans and lakes. It is also employed in quality control to measure the concentrations of sugars, salts, or other solutes in food products and beverages, ensuring consistency and flavor. It is also applied in research to study the properties of various solutions and to determine the qualitative properties of different solutes and solvents. It is also used in cryopreservation to measure the concentration of cryoprotectants in biological samples like cells or tissues that are being preserved at very low temperatures. In environmental science, used to check the purity of water by measuring the freezing point depression caused by dissolved impurities or contaminants. Let's look at its advantages and disadvantages. Advantages are it is highly precise for concentrated solutions. It provides accurate measurements for solutions with high solute concentrations such as concentrated salts or other non-volatile solutes. There is no need for calibration curve for colloids. It is it is also reliable for non-volatile solutes. It provides high accuracy in determining osmotic properties for systems with high osmotic pressures. Disadvantages are freezing point depression takes longer to measure as it requires cooling the solution and waiting for the freezing point to stabilize. It needs relatively larger sample sizes compared to other osmometry methods, which can be impractical for limited or precious samples. It requires precise temperature control to ensure accurate freezing point measurement. and fluctuations can introduce errors. Impurities in the sample, such as non dissolved solids, may interfere with accurate freezing point determination. It cannot be used effectively with volatile solutes like alcohols, as the solute can evaporate and alter freezing behavior. Now, moving on to the vapor pressure osmometer, it measures the lowering of vapor pressure in solutions. Now, how it works. Two meshed thermistors are placed in a sealed chamber saturated with pure solvent vapor. The chamber is maintained at a stable temperature to ensure consistent vapor pressure conditions. A drop of pure solvent is applied to each thermistor. Both thermistors equilibrate to the same temperature since the vapor pressure around them is uniform. One thermistor, reference thermistor, remains coated with the pure solvent. The second thermistor or sample thermistor is coated with a drop of the solution whose concentration is to be measured. The vapor pressure of the solution is lower than that of pure solvent. As the chamber is saturated with pure solvent vapor, condensation occurs on the sample thermistor to equalize the vapor pressure difference. The condensation process releases heat, causing the sample thermistor to warm. The temperature change causes a corresponding change in the electrical resistance of the sample thermistor. The reference thermistor remains unchanged, serving as a stable baseline. A bridge circuit detects the difference in resistance between the two thermistors. The resistance difference is proportional to the temperature difference, which reflects the vapor pressure depression. The resistance change is converted into an electrical signal by the bridge circuit. This signal is then sent to a display unit, for example, a panel meter, strip chart recorder, or computer terminal. The recorded signal is compared to a pre-calibrated curve that relates resistance changes to molar concentrations. The concentration of the solution is determined and displayed. The system is reset by cleaning the thermistors and chamber to prepare for the next measurement. Now, these are its applications. It is used in the measurement of osmolarity in biological fluids such as blood plasma, serum, and urine. It helps in diagnosing and monitoring conditions such as dehydration, hyponatremia, and hypernatremia, and other electrolyte imbalances. It is used in critical care to monitor the osmolarity of solutions administered to patients, ensuring they are safe for intravenous use. It determines the osmolarity of injectable drugs, eye drops, and other medications to ensure they are isotonic and safe for human use. It is used to determine the molecular weights of polymers, peptides, and small molecules in pharmaceutical research. It provides information about the stability of formulations by measuring changes in osmotic concentration over time. It determines the osmotic properties of proteins, nucleic acids, and other biomolecules. It helps in understanding their behavior in solutions such as aggregation, dissociation, or hydration. It measures changes in osmotic concentration during enzymatic reactions, helping to study enzyme kinetics and the substrate binding. It also ensures the consistency and quality of solutions used in food and beverage production, cosmetics, and industrial formulations. It was also used to measure the concentration of sugar, salt, or other solutes in food and beverages. It ensures the correct balance of osmolarity for taste, preservation, and safety. These are its advantages and disadvantages. Advantages being it provides quick and real-time measurements based on vapor pressure differences, making it suitable for fast analysis. It requires very small sample volumes, typically microliters, making it ideal for limited or precious samples. It offers high accuracy and precision, especially for solutions with low osmolarity, making it perfect for clinical, pharmaceutical, and research applications. The sample is not consumed during the measurement, allowing for reuse or further analysis. The disadvantages are this method becomes less sensitive when the solution concentration is too high, as the vapor pressure difference between the solvent and solution can become negligible. It is best suited only for the volatile solvents and may not be applicable for solutions where the solvent doesn't vaporize easily, such as highly viscous or non volatile solvents. Its accuracy can be affected by temperature fluctuations as the vapor pressure is highly dependent on temperature. Any temperature variation must be controlled very precisely. The device requires accurate calibration to correlate resistance changes in thermistors with osmolarity or concentration. And this calibration curve may need adjustment for different solutes. The device can be very expensive and may require regular maintenance, particularly to ensure the precision of the thermistors and the overall system. Moving on to the membrane osmometer. It measures osmotic pressure using a semi-permeable membrane. The semi-permeable membrane here separates two chambers. One chamber is filled with pure solvent, for example water, and the other is filled with a polymer solution or any solution containing solute. The solvent chamber is sealed except for the membrane that allows solvent molecules to pass through. The solute chamber is left open to the atmosphere, meaning the solute side is exposed to air and can exchange with the surroundings. In the pure solvent chamber, the chemical potential of the solvent is higher because the solution is undiluted and has a greater tendency to move toward areas of lower concentration. The solute is present. In the solute chamber, the chemical potential of the solvent is lower because of the presence of solute molecules. The solvent molecules are less free to move due to the interactions with the solute. Due to the chemical potential difference between the solvent and the solute chambers, the solvent will naturally flow from the solvent chambers where it has a higher chemical potential through the semi-permeable membrane into the solute chamber where it has a lower chemical potential. This process, called osmosis, continues until the concentration of the solvent on both sides of the membrane is more balanced or until the osmotic pressure equilibrates. As the solvent moves into the solute chamber, it dilutes the solute and increases the volume of the solute chamber. This increase in volume leads to an increase in pressure inside the solute chamber. The osmotic flow of solvent continues, causing the pressure to build up until the pressure difference between the two chambers reaches a point where it counters the chemical potential difference caused by the solute. Eventually, the flow of solvent slows down and it stops when the pressure exerted by the solvent in the solute chamber equals the difference in chemical potential between the two chambers. The pressure required to stop the solvent flow is known as the osmotic pressure. This is the pressure that exactly balances the chemical potential difference, thus preventing further solvent diffusion. The osmotic pressure is measured by the pressure sensor or manometer attached to the solute chamber. This pressure is a direct measure of the osmotic pressure that has built up due to the solvent diffusion. These are its applications. It is used in molecular weight determination using the Van 't Hoff's law. It is used in biological studies, pharmaceutical and drug development, food industry, water treatment, environmental science, quality control and testing, and nanotechnology. Nanomaterials like carbon nanotubes and graphene oxide are being researched for use in membranes that exploit osmotic pressure for more efficient water filtration and separation process. These are its advantages and disadvantages. It measures osmotic pressure directly, which can be used to determine the concentration of a solution. It is relatively simple in operation and often more durable than other types, requiring fewer moving parts and less maintenance. It does not need pre-calibration with known solutions, which simplifies the procedure and reduces errors from calibration. It works effectively with a wide range of solute concentrations, including moderate osmotic pressures. It is particularly effective for non-volatile solutes, where vapor pressure and freezing point diffraction methods may not work well. Uh, Disadvantages: the system may take time to reach osmotic equilibrium, particularly with very low or high concentration solutions. It may not be very accurate for high osmotic pressure solutions, as the osmotic pressure can exceed the capacity of the membrane to measure accurately. The semi-permeable membrane must remain clean and free from blockages. Contamination can significantly affect measurement accuracy. The type of membrane material can limit its applications to certain solvents or solutes, particularly if the solute molecules are too large to pass through the membrane. It is also limited for colloids and large molecules. In conclusion, osmometers are essential instruments that measure osmolality by taking advantage of colligative properties such as freezing point depression, vapor pressure lowering, and osmotic pressure. These devices are invaluable in clinical diagnostics, pharmaceuticals, biotechnology, and chemical research, with each type tailored to specific application needs. As technology advances, osmometers are poised for significant innovation. The future will bring smaller, more portable devices, increased automation, higher sensitivity, and the potential for real time continuous monitoring. These improvements will make osmometers even more accessible, efficient, and precise, opening new possibilities in diagnostics, research, and quality control across industries. By keeping pace with these advancements, the field of osmometry will continue to provide valuable insights into the molecular composition of solutions, supporting progress in healthcare, pharmaceuticals, and scientific research. Thank you.